So I like that introduction of we couldn't have said it better, so Scott's going to come up and try to say it better. And I won't say it better. That was a wonderful introduction. So I'm going to try to, you've heard a lot about this platform trial. By the way, my name's Scott Berry. I'm a biostatistician working on the design of this. I'm going to try to explain what is this thing? What does it look like? We've heard of this platform trial. What does it look like? And sometimes the best thing, you can skip the reading and I'll just show you the movie. So you can watch the movie. I'll show you the movie of what this looks like. I think that may help. I'll address goals of the trial and how does this potentially address these goals we're all talking about in terms of the trial. So there's been a lot said about drug development and some of the difficulties, the startup costs, the creation of sites, IRBs, your data safety monitoring board, the long delay, and lots of numbers thrown out, but approximately 2 years to get your trial started. You enroll the trial, you enroll 1-to-1, and then you shut the trial down. You collect the data, you do your analysis on this. The landscape of multiple drugs doing this in silos, where there's drug A, there's drug B, there's drug C, a patient in the trial for drug A doesn't contribute to drug B. That patient is separate, that patient's enrolled in a different protocol, different inclusion-exclusion criteria. This is a little bit of the apples and oranges and pears that are discussed. So each one of these becomes its own inferential discussion of did drug A work. There's a lot of white space that happens here as well. There's a lot of planning and a lot of shutdown that goes on in this setting. So what we'd like this to be is this is what a platform trial looks like. We don't, in this setting, we'd like to have multiple arms come in within the same arena on this. They play by the same rules. Patients come into the trial and arms come and go. It's all within the same infrastructure. The same protocol, the same standardization of endpoints. Arms come and go, and this as Billy described, this perpetual living trial continues on as long as we continue to have questions. The hope is we take the previous slide from this, where we have this, and the development looks more like this. Yes, startup and creation of this is longer. It takes longer to create a platform trial than it does a single trial. It's not too much longer, actually. It's a little bit longer. And then we have multiple arms in the trial simultaneously. And then new arms come. These arms leave and new arms come in during the course of the trial. It's the same protocol. A patient enrolled in the middle of this or the end of this is enrolled into the same trial. A patient that's enrolled into, in the upper left, into drug A, that patient's data is continually used as more arms come into the trial during the course of that. It's not a separate entity that's locked away and gone. It's a common arena that we can continually address questions. As this goes on and we move off to the right, you can see we're comparing to arm D. The arms to the right are comparing to arm D. That was in previously that we found was safe and effective. It's now being used as the standard of care, and you may have placebo on top of standard of care, but now we're adding things to D and we're comparing to that in this living trial that continues on. So this is what we hope the platform trial to look like. So there's one master, you've heard this term master protocol. There's a single master protocol that drives this trial. Arms come and go as appendices to the trial. When that arm is done enrolling or its exposure in the trial is done, the appendix is removed in a modular fashion and the trial continues on. The patient experience is standardized. The visit schedule, the endpoints that are collected are identical across all of the arms. Now you see the ability and the synergy to use patient data from one to the other because the experience is exactly the same. The endpoint collection is exactly the same. All of the data is being used for all of the arms. All right. So the goals of this is not a zero-sum game. Ooh, we're doing something effective. It must be worse data for the FDA. Or we're collecting better data for the FDA. It costs more. It's more burden to the patients. What we're trying to do is this is better for all of the stakeholders that we've been discussing. The goal is to treat patients better, those in the trial and those patients yet to come, that we get better treatments and we're treating the patient in 5 years and 10 years better, but also the patient today. It doesn't have to be a negative for the patient today, for the patient tomorrow. We want to improve the efficiency of drug development. We've heard a lot that this can be done cheaper. A single sponsor coming into this trial will cost less than if they go off and build an arena themselves to conduct the experiment. It can be done faster. We can be first patient in and we can get our data faster. We can collect a smaller sample size with more strength, and that's faster as well. We can enroll a wider patient population, and Deborah is going to talk about the inclusion-exclusion criteria during the course of this, that it doesn't have to be just this very narrow region of boys that are about to decline on 6-minute walk. We can do it a much wider range of the patient population. And all of this together can accelerate finding therapies that are effective. So here are some details of the trial, and then I'll show you the movie of the trial. So this is the idea here is that a patient comes in and their inclusion-exclusion criteria in the master protocol. The patient becomes eligible for the master protocol. At this point, there isn't even an intervention that's specified on that. Remember, interventions come and go in a modular fashion. When a patient is then eligible for the master protocol, they are randomized into, and these are the red arms here, they're randomized into the different arms that are enrolling patients at that time. And here's an example where A, B, and C are enrolling. So a patient comes in, they're eligible for the master protocol, and Deborah's going to talk about the inclusion-exclusion for the master protocol. Then they're randomized to the different appendices. Their patient experience of data collected is the same in each one of these. Of course, the treatment given is different. Once randomized into one of these appendices, the patient is not blinded across A, B, and C. They're only blinded within A, B, and C. They are randomized 3-to-1 active to placebo. In the bottom for arm C, it might be that there's actually 2 doses given for that drug. And the 3 is split into 1.5 and 1.5, but it's always 25% for placebo for each one of these. When A is making inferences in this, they are using placebos from the other arms. It's a randomized comparison with the same inclusion-exclusion criteria. This is where multiple arms working together, the placebos across here become comparable, and they can use those in those decisions. In the 3-to-1 randomization, in one experiment, if you do 3-to-1, you reduce the strength of that comparison because one arm is smaller. Here, adding them together, you don't reduce the strength, yet we enroll smaller to placebo. So this is what it looks like both in an arms experience. I'll say a little bit more about an arm coming into this. What's the experience of an arm? But you can see the master protocol setup. So here's an example. Let me back up a little bit. And then I'll show you the movie. I keep promising the movie. The potential primary analysis, one of the nice things of this is you can analyze the data within yours using placebo from other arms, using different analyses. It doesn't force everybody to do the same analyses. Now, we'd like to, the scientific information that comes from all of the data in this, and for example, Jane's work in mathematical models of these analyses, we can improve this as time goes, how we analyze the data. Here is an example analysis that allows a much wider range of the patient population to be included. Here are 2 endpoints, the North Star ambulatory measure and the PUL, which is an upper limb motor test. This might be what the patient's experience looks like, where initially the North Star decline, ambulation, loses ambulation, and then the upper limbs are declining as well. If we institute a treatment at different times during the course of this, early on in the disease, the dashed lines show a 50% slowing of the rate of decline on both endpoints simultaneously. In the upper left, you're not going to see a difference in the upper limb measure, but you will on the North Star because that's what's starting to decline, and you can see the red line difference between a placebo patient in solid and a treatment that slows decline 50%. In the middle, you're going to see that on both. And at the far right, you're only going to see that on the pole because the ambulatory appendix is essentially zero at that point. And you could imagine even adding in endpoints, FVC or some other endpoints on the farther right of this. But this incorporates, and you gain strength from every patient on the wide range of disease, analyzing, does the drug slow the rate of decline, using two endpoints jointly in that analysis. So we're using that in this movie. This is a demonstration of what this may look like. On the top part, it's going to show the enrollment of 4 arms. And it's a perpetual trial, but I have to end this movie at some point. So I'll show you the first 4 arms in the trial. It starts off with red enrolling for a short time, and then blue starts enrolling. At the top, you'll see the cumulative number of patients enrolled, and then in the bottom left, you'll show the maturity of each arm's data, and in the bottom middle is the percent slowing parameter. This is all simulated data, a simulated movie. I'll move it to 9 months in the trial. You can see blue and red are enrolling. They're jointly enrolling each of their placebos, which are combined together in the black line. Now, the maturity in the bottom left, the data are maturing. It says weeks there, those are months. We're starting to get mature data on each of those arms. In the bottom middle shows that initially an analysis of that, an interim analysis, would show that those arms are slowing the rate of decline. If those arms move entirely above those dashed lines, it's starting to get into statistical significance. If it's staying below or staying near those lines, we're going to stop the arm for futility. This moves on to this point, and the blue arm stops for futility. Its confidence interval for its treatment effect is not separating, where we have a certain amount of confidence that blue is not effective. Those patients are going to be removed from that arm at that point and re-randomized into a new arm. There'll be multiple experiences for a single patient. Now they can move on, and you see purple has added the trial. The purple arm had started enrolling. Red is still enrolling, and we've actually stopped enrolling, and we're following the red arm. The red arm looks like it has about a 20% slowing, but it's unsure at this point. The green arm has enrolled. We're now starting to enroll green and purple at this point in the trial. And you can imagine now multiple arms continuing to go. Blue stopped for futility. Red was unsuccessful in the trial, but now purple and green are going. And of course, now there'll be more and more colors entering the trial, where all of the data are being used now for each individual arm's analyses. Now, purple is successful and green are successful. And you can imagine multiple colors going into this. Now, a patient enrolled at 6 months would have been randomized between red and blue. A patient enrolled at 15 months would be randomized to green, purple, or red. A patient on the blue arm would have washed out and could be re-randomized to red, purple, or green for that arm enrolling. The patient may have multiple experiences through the course of this. That actually creates more statistical efficiency when you have the longer length of data for any individual patient and their exposure across the arms. Okay, so that's what it looks like. I talked a little bit about the patient experience, a wider range of patient. The randomization across appendices. This is different than what you're probably used to when there's only drug A. It's placebo or A. Now you can be randomized to A, B, or C, and then within there you're randomized to its active or its placebos. The 3-to-1 is across all arms. That's mandated by the master protocol. And then the re-randomization if that arm is found ineffective. If that arm is found effective, each patient is moved on to a cohort study, and they're continually followed on that arm. They stay on that arm into an open label extension. If not, they can be re-randomized, washed out and re-randomized. Intervention owners, they have to maintain the 3-to-1 randomization. The hope here is that the protocol's already running. Patients are being enrolled during the course of the trial. The time to first patient is faster. You can do the 2 years of starting up your own trial, or here you can get in much faster than that. Hopefully about 6 months from, okay, we're ready to go, rather than 2 years. It's good for intervention owners. Hopefully this becomes, if there's one arm that enters in this trial, it's going to be more expensive, but when multiple arms enter into this, it becomes cheaper. You enroll fewer placebos, but you get the strength of, as though you enrolled more, from borrowing across the arms. The negative, if there is to an intervention owner, is the inclusion-exclusion criteria are set. People have to maintain each of those. We're working with multiple sponsors, multiple organizations to create a nice standardized set of inclusion-exclusion criteria that's quite broad that will be acceptable to multiple intervention owners. It's fit for phase 2, it's fit for phase 3, it's fit for phase 2-3 seamless trials. Dr. Dunn talked about that, the ability in the trial to be looking at biomarkers and advancing to phase 3, all within the same trial for an intervention owner. All right. Some of the advantages of this, I'll ignore the dash lines here. The solid lines at the top show in red a single intervention owner that enrolls 3-to-1. That's what the power curve looks like. The sample size across the x-axis. The y-axis is the power to demonstrate a benefit if the intervention has a 30% slowing. The green line shows if 2 arms enroll simultaneously, 3-to-1 each, and they each use the other's placebo. There's this increase in power that comes with it with the same resources. And then the blue line at the top is 3 intervention owners simultaneously enrolling 3-to-1 in the same platform trial using each other's placebos. The sample size needed for 80% power for the exact same resources goes from about 125 to 75 for that intervention, and all they've done is enrolled in the same trial. So there's immediate advantages of each of the arms working in the same protocol. Oops. So imagine 10 therapies out there, and we saw all the available therapies. They're all working in individually, solo development, 1-to-1 randomization. They're using this more innovative analysis, the same analysis in both. They enroll 75 versus 75 in a trial, 80% powered for a 30% effect. Meanwhile, suppose they all enrolled in the platform trial and they enrolled 75 to 25. They have the same power in that setting. They enroll 1,000 patients rather than 1,500. They enroll 250 placebos rather than 750. So they save 33% patients right off the top. 50% reduction in placebo patients enrolled during the course of this, yet each arm has the same strength. It decreases the time to find a successful drug across the 10 by more than 50%. Each one of these are shorter. Each one of these have the same strength. This also ignores a great deal of things that Dr. Dunn talked about. The disease learning by having 10 drugs all in the same database to hand to critical path to analyze, the exact same endpoints and all of this, moves the disease faster than the silo development where the data are not shared. We have to create consortiums to share that data. Here, it's all together in one setting. The I-SPY 2 trial is a neoadjuvant breast cancer trial. They've had 15 drugs go through that trial. All of the data is in one case. We're looking at surrogate markers that you could never look at in a single drug. How do 15 different drugs affect the tumor markers, and what does it mean for event-free survival? It's unique in the settings. This could happen in Duchenne. So all of these advantages are available, with many other advantages that come with all of the arms working in the same framework. So some of the things that are new and different in this are quite a quote-unquote controversial. Randomizing across appendices. This has been a good bit of discussion of what that means to patients. Is that positive, negative? But it's different than a single trial. It's a new thing in platform trials. The re-randomization of a patient in the same protocol and having multiple experiences across interventions. A much larger inclusion criteria across this. Again, this is a trial built by patients, for patients. To the benefit of arms as well. The analyses of these across the wider range. You can't look at a single endpoint when you're enrolling patients from sort of 4 years old or 5 years old to 15 years old. Different endpoints matter across that stream. How do we analyze them simultaneously? As a statistician, of course, we enjoy this part of this challenge of trying to do this. So the other part is when an intervention comes in, they can't come in and say, I only want these patients for power purposes. They can restrict the patients for safety, for sure. If there's a safety aspect of it, they're not going to enroll those patients. Or if it's mechanism of action. If the drug doesn't have a potential to benefit a particular patient, that patient won't be eligible for that arm across the randomizations. And then the borrowing would be across only patients eligible for that particular intervention. And we can track that in the same trial. We're starting off with the wide range of this. Dr. Dunn talked about the ability in a trial to do precision medicine, and that's absolutely part of the plan, but we're not set up for the first arm to be an exon 51 skipping gene therapy where only 8% of patients are eligible. We want to hit the large set of this, get multiple arms started, and then we can start to enroll the subsets during the course of the trial much easier. It would be hard as the first arm. So we're delaying that part, but it's absolutely part of the plans for the Diane TU trial, for the Duchenne trial. All right, I will stop there. Thank you.